Hi, folks, and welcome to part two of lecture two. Um, I just wanted to give you a heads up on what is coming up. So it's about 55 minutes plus one this minute, probably. Um, so in the beginning, uh, it'll continue directly from where it left off in part one, which was basically just one long lecture, almost 2 hours. I've chopped it up a little bit and changed things around, okay? But it will start summarizing part one, trying to give context between specification and types of development, bringing in terms we've used, mosaic and regulative. And we will try and make a timeline of events with the terms that we have learned, and we see that the times these things happen at are very overlapping and it's difficult. We will go into start discussing the term induction and different cues, trying to explain that, not different cues, different responses, and how the capability to respond to this signal is referred to as competency. Then we will take a couple of signaling pathways, and throughout the sort of beginning part of the course, we will bring in signaling pathways and discuss them in some detail. So these Wind and Sonic Hedgehog we'll start discussing, and maybe the delta notch pathway actually comes in a bit later on. Then we will go back using terms we know in induction to discuss patterning, and I will introduce the term morphogen and morphogen gradients. And this will take about 50 minutes in total. And then the last part, it's kind of a standalone part almost, looking at animal models very quickly. I will go through 4 or 5 animal models using about 1.5 minutes each. And these are sort of relevant to where we will meet them in the next lectures in more detail, right? Possibly without the introduction to them. Okay, let's get started, shall we? So we could demonstrate that there was specific cell states at different developmental stages, and that with time, the cells in the embryo become more restricted in their developmental potential. And this was true whether the embryo had a more mosaic or regulatory style of development. A mosaic, as again I can mention, is where they had these, the model has these nuclear determinants that are shared unequally between cells as they divide, giving rise to new identities after division. And regulatory was more after a division, the cell has the possibility to become one type of cell or another, and its interactions between the cell surroundings that will give rise to their choice of fate or influence their choice of fate. In both mosaic and regulatory cases, these fates could be maintained over time in several rounds of division, or they could become further restricted as the cell proceeds more towards a differentiated state. And I just mentioned here that these mosaic style cleavages are very similar to asymmetric cell divisions that we will hear about later on, where the determinants are unequally distributed in the cell, and as the cell divides, it passes the majority of these determinants to one daughter cell. We can briefly restate the differences between regulatory and mosaic development. Development of more regulative embryos depends on this cell-cell interactions, while the mosaic embryos depends on cytoplasmic determinants. And there is, as I said, a spatial mosaic of molecules, which can be mRNAs or proteins within the egg cytoplasm, and that these are unequally distributed to different cells during the cleavage, which gives rise to daughter cells that have distinct identities. And this usually leads to an early determination of cell fates in the embryo. But it's worth noting that purely mosaic embryos do not exist, so there is no animal that we know of that just uses this mode of development. Mouse embryos, then, on the other hand, are more regulative. They're capable of considerable regulation, and this can be demonstrated by mouse chimeras, where and a chimera is a mixture of cells that are genetically different. We can take from 2 different mouse strains at the 4-cell stage and put them together, and this will give rise to a functional adult composed of both cell types from the different animals. And this shows how regulative it would be. But because if this was mosaic, where it was already determined at this stage, we would expect perhaps we get two animals out, right? The cells don't interact at all. But here they fuse together into one embryo and regulate each other so that you get one normal sized and proportioned animal. And these mouse chimeras have been analyzed to test whether certain genes are important and act in a cell autonomous or non-autonomous way. And I mentioned these definitions before. And cell autonomous is the gene that only has effects on the cell that it is expressed in. Typical example of this would be it's a transcription factor. It's not secreted across to the neighboring cell. It's acting cell autonomously. A non-cell autonomous gene affects the cell other than the cell in which it is expressed. So, for example, if it was to encode a secreted signaling protein. These are the strict definitions of whether something acts cell autonomous or non-cell autonomously. If we try to sort of put these processes together, or specification and determination in terms of the embryonic processes that we've described, like the cleavages and the patterning followed by gastrulation, we can see that specification happens usually early on. And of course, this will depend on whether it is mosaic or regulative, so type of embryo or development. The mosaic style will specify and determine cells early, whereas the regulative type of embryonic development will kick in with specifying and determining cells later. It's hard to give a defined determination starts at this phase. It's sort of, there are differences between tissues and organs. So that's why there's no clear map of this. There's an overlap between these processes, right, of determination and the movements and patterning that happens during embryonic development. And then, but we can agree that sort of terminal differentiation occurs much later, and then growth is kind of the last phase to happen in the animal, where basically you have a fully developed animal, just needs to grow in size. So what do we mean when we say it's dependent on cell-cell interactions and influences from other sides? So here a term to consider is very important, and that is induction. So induction is the signaling from one group of cells that affects how another group of cells in the embryo will develop. And a very important inducer is called the Speeman organizer, and it is found, shown here, in the dorsal lip of amphibian embryos, and it acts as a very important inducer of other tissues. Inducing signals may be propagated over several or many cells, or may be highly localized, so acting on a 1 or 2 cell distance, for example. There are a number of different signaling centers or organizing regions, as they can be called, and these are established in the embryo at very specific locations at certain times and stages, for example, in the developing brain. Importantly here, in terms of learning terms, is that there are 2 different types of inductions that can be distinguished. There are permissive inductions, where a cell can only make one kind of response to that signal. So that signal will only give an on or off response from the cell that it's trying to induce, or induce to become a certain cell fate. So, and this requires enough signal and that the receiving cell is competent to respond, right? We'll explain this now in a bit more detail as we go on. The other type of inductive cue is instructive. And instructive inductions occur when a cell responds differently to different concentrations of the signal. It could also be thought of as being selective, that the responding cells has limited possibilities which the instructive cue can select between. So in this case, there is only one kind of response. It's kind of on or off to receiving a signal. But in the instructive case, there are different fates that are possible for that cell in response to the same cue. And there's examples here to help us understand the difference. In the permissive induction, we have this figure here where we have a frog gastrula and a newt gastrula, and we can do transplants between these animals since they're so close. But these animals normally look in a certain way. If we have the frog here, he normally has these suckers placed here, and vice versa. The newt here normally has these kind of balancers under his mouthpiece. Now these need a cue to develop and form these structures. And if we take the frog mouthpiece area that we know is specified to become a frog mouthpiece and we transfer it into the newt gastrula, it is induced to form these mouthpieces, but it's only forming the frog mouthpiece, right? So in a way, you could say it was already determined to become this. But the other way is that the signal that comes to induce the formation only has one possibility. So this ectoderm, it only knows how to make this kind of a mouth, but it needs an inductive signal to do it. But the inductive signal cannot tell it which type of mouth to become, only to become a mouth. In the case of instructive, the cell can respond differently to different concentrations of the signal. So in this case, if we take an ectodermal layer of a chick, which is not specified or determined yet, and we introduce it to signals from a mesoderm, depending on the strength of that signal or where it's coming from. So if we introduce the wing ectoderm to wing mesoderm, the inductive signal will cause the wing ectoderm to form a feather, which would be the correct thing. But if we take some thigh mesoderm and introduce it to the wing ectoderm, it will cause the wing ectoderm to develop into a thigh feather. And if we take some foot, it would cause the wing ectoderm to develop into scales and a claw, like on a foot, right? So this wing ectoderm can respond in different ways to instructive induction cue. So there is more plasticity. It's less specified in the wing ectoderm, and it can respond to different strength of signal by giving different output. So the cue itself then is said to be instructive. We should also mention that in development we have antagonistic or negative signal molecules that can block induction by preventing the inducing signal from reaching the cell or binding to the cell surface receptor, for example. It's important to keep in mind. So these inductive signals, as I said, they can pass over larger distances or quite short distances from cell to cell, and they move by different mechanisms. And diffusion is one mechanism. We see an example of it here, where you have the secretion of a ligand, for example, to the neighbor cell, and depending on how capable of diffusion this signal is, it will travel short or long distances. And examples of this would be the secreted diffusible molecule like TGF-beta or Wnt, wingless signaling ligand. And the typical thing then, it would be binding to a cell surface receptor and in causing a signal transduction event in the receiving cell, inducing it to differentiate, let's say. We can also have sort of direct contact between cells, where we have ligand and receptor bound on the cells, for example, the delta notch signaling, which is very important during development. And then again, the contact between the two cell surface receptor ligand causes a signal in one of the cells with the receptor that is important for inducing cell fate. But also some small signaling molecules can travel through gap junctions. These are proteinaceous structures that join cells on the lateral membrane, and small signals can pass from one cell to the other, and we can have direct communication between the cytoplasm of adjacent cells, and this is one way in which an inductive signal could be sent. We can mention that there are other ways, like small hydrophobic molecules, for example, retinoic acid. It can travel through the cytoplasm and penetrate plasma membranes, binds to nuclear receptors to cause transcriptional changes. And there, in recent times, it's become more and more appreciated that there are other mechanisms, and these include cytonemes and exosomes. And I just thought I would mention these very briefly, but they won't be on the exam. So just thought it was interesting to show you that cytonemes, we see them here, they are projections from cells. There are different types of projections that either bring an actin-based protrusion close to another cell, and that can be enough that they just bring, for example, a ligand that is membrane bound to another cell sitting here to receive the signal. And this is some examples are shown here. So here we have a cytoneme from this cell that has ligand on its cell membrane, and it can reach a cell several distances away that has the receptor for it and cause changes there, including inductive signals. And there are other examples of this given here. Here in this picture, we can see they've used Life-Act, which is a stain. Remember, we mentioned these vital dyes that can stain living cells. Life-Act is one of them. Binds to actin inside the cell and fluoresces. And then they have cells that are also expressing an M-cherry tagged protein to mark these cells. And what they see is that when a cell sends out a protrusion, the red cell sends out a protrusion and contacts the non-red cell, which we can see the green from the Life-Act here. We can actually see that it can pass some red protein directly to this cell. So, and this is kind of an open-ended actin-based protrusion. They have different names, but basically it can deliver different signal molecules from one cell to another cell over several cell distances. And I just thought you should mention at least exosomes. They are an important way of secreting Wnt signals, and Wnt is very important in development. And I don't know if you've heard about exosomes before, so it's interesting for some people. It's quite a hot topic at the moment. Basically, endosomes are internalized into the cell with molecules. For example, it could be the Wnt ligand. They are packaged and sorted into another vesicle inside the cell called the multivesicular body. And sometimes this multivesicular body fuses with the plasma membrane and releases vesicles with signaling molecules on them. And these are called exosomes. And these vesicles can then travel throughout the body, large distances, and bind to cells and be internalized and deliver signals that way. That's something that if you're interested in, you can have a read about it, follow up yourself. So we mentioned signal transduction many times, and that we should have a refresher on how this is working. Basically, you have the binding of a signal protein to the extracellular face of its cell surface receptor, and this causes signal transduction in the cell, which is an activation of a relay of intracellular signaling proteins that results in a cellular response. And here we see an example of a receptor with the proteins inside in the cytoplasm. It's not activated at this stage. Along comes the ligand. Particularly relevant to this model is FGF, fibroblast growth factor, or EGF, epidermal growth factor. These are our growth hormones, and they act by binding to the receptor. They cause transphosphorylation on the intracellular side. This activates these proteins. It recruits and activates these proteins, triggering a phosphorylation cascade. Typically would be this MAP kinase pathway, if you've heard about that. And then one of the terminal proteins, which is usually a kinase, at the end of this phosphorylation cascade, translocates into the nucleus where it phosphorylates a transcription factor, and this gives target gene expression. But there are other models, for example, signaling resulting in the movement of gene regulatory protein into the nucleus. So in this case, which is relevant for the the hedgehog, Wnt or wingless, and TGF beta signaling. We have a ligand binds to the receptor. Inside in the cell, we have a signaling complex that is, for example, held inactive normally, but when the ligand binds to the receptor, it triggers changes in the signaling complex that releases the transcription factor, which can now translocate from the cytoplasm into the nucleus and trigger transcriptional changes. And we have another class where the activated receptor is actually cleaved and translocates itself or a fragment of it into the nucleus and acts as a co-activator. And that's particularly important in delta notch signaling, which is particularly important in terms of inductive signaling. Here we see that the delta ligand on one cell contacts the notch receptor on the other cell. This interaction triggers the cleavage, proteolytic cleavage of the intracellular domain of the notch receptor, and this then can translocate into the nucleus and act as a co-activator for transcription factors and trigger target gene expression changes. We can have a closer look at the Wnt signaling pathways. So first homologue of Wnt was discovered in Drosophila. Its phenotype was a wingless flies. So the abbreviation for the fly mutant would be WG, wingless, and it's called Wnt wingless related proteins then in other species, including in human. And Wnt is a receptor ligand signaling mechanism. It can activate more than one signaling pathway. The two common pathways that are referred to are the canonical pathway and a non-canonical. So the sort of the normal and the non-normal. And the non-canonical pathway is involved in cell shape changes, and these can be important for migration, particularly in cancer cases where we have changes in expression of Wnt proteins, and this can encourage the cells to start migrating and metastasizing. Right? So in this non-canonical pathway, Wnt is the ligand that is secreted from cells. It recognizes the frizzled receptor, binds to it, which activates a protein downstream called disheveled, which activates a protein called DAM1. And DAM1 can activate Rho-A, which is small GTPase involved in cytoskeleton remodeling. And this acts through ROCK2 as a kinase that can activate cytoskeleton reorganization. Additionally, the non-canonical pathway via disheveled activates J and K, which is in the MAP kinase pathway, which activates a transcription factor. So it activates several pathways, the non-canonical pathway, and it will be in certain cells. The canonical pathway is set up, it's a little bit more traditional looking. We have the receptor, a co-receptor, and again, the disheveled protein that is important. And this pathway signals through beta-catenin. Beta-catenin is a protein that is found in adherence junctions at the lateral membrane of cells, but also it's in the cytoplasm where it can act as a signaling molecule. And in the inactive case, when there's no Wnt signaling, the ligand is absent, beta-catenin is bound by a set of proteins called the destruction complex, which is made up of APC, axin, glycogen synthase kinase 3, and CK1 gamma. And these bind to beta-catenin, phosphorylate it, and trigger it for degradation, right? But when wingless is bound to its ligand, its co-receptor is recruited, we get phosphorylations on the co-receptor and on disheveled that lead to a recruitment of the destruction complex to the membrane and free beta-catenin in the cytoplasm, or allow it to build up in the cytoplasm, where beta-catenin can translocate across the nuclear membrane to the nucleus and activate a transcription factor called TCF, leading to transcriptional changes. And TCF, when in the absence of Wnt signaling, is held inactive by repressor complexes, HDAC and CTBP and Groucho. Okay, that is canonical wingless signaling in sort of a 2-minute version. So another important pathway to discuss in development is the sonic hedgehog pathway, SHH.
And again, this was first described, the sonic hedgehog mutation was first described in Drosophila, where it was called hedgehog because of the appearance of the embryo. So I hope you noticed that, well, first of all, flies discovered a lot of these important proteins for development. They are also important for cancer. I've mentioned that before. And that the other thing is that the phenotype in the fly often reflects its name. So hedgehog has this look. Wingless was without wings. I should say hedgehog. I think it's called pinsvin in or is it egelkott? I can't remember now, in Norwegian or Swedish. It's a small spiky mammal. Is the hedgehog. Yeah, and this is the normal embryo, what it looks like. But when they found the mutations in hedgehog in a recessive screen, the embryo looked like this. We don't have time to really go through this, but I wanted to show you that in the sonic hedgehog pathway, many of the phenotypes from the key players in the sonic hedgehog pathway, many of the phenotypes are related to how hedgehog look, affecting this bristle formation on the outside of the embryo. But we don't really have time for it, but I will mention how the sonic hedgehog pathway works. We have a ligand, sonic hedgehog, which binds to the patched receptor, and the patched receptor normally functions in the absence of ligand. The patch receptor functions to block the activity of another protein on the membrane, smoothened. Again, notice the name. You can imagine what the phenotype looks like in the fly. Is there smoothened here? Oh, never mind. Normally then smoothened would hold the transcription factor Gli out of the nucleus, but when sonic hedgehog binds to patched and smoothened is inhibited, then smoothened no longer blocks the translocation of the transcription factor into the nucleus, and we get activation of transcription in response to sonic hedgehog ligand. And sonic hedgehog protein regulates many developmental processes in humans and vertebrates, as well as in Drosophila. But it was identified as one of the genes affected in patients with inherited holoprosencephaly, which is characterized by midline brain and facial defects. They found that the plant alkaloid cyclopamin inhibits smoothened activity, and through a lack of smoothened activity, we found that sheep that have eaten plants that contain this cyclopamin, they have these very severe facial defects. They're sort of cyclopea. They're almost like a cyclops. This just to illustrate that these pathways we're talking about are indeed important in development. So we talked about induction. That one part was the signal, was very important, but also how the cells had to be competent to receive the signal. Not all cells will be competent for that, and what this depends on is how the cells have been programmed up to that point. Let's say what signals they have met. So the cell can respond to these signals coming in by modifying enzymes or cytoskeleton. We talked about phosphorylation events that lead also then to alterations in gene expression, so transcriptional changes in the cell, gene activation, or in some cases it could be repression. Yeah, and we have other types of signals that can activate transcription factors that are also nuclear receptors. Yeah, but what about the competence of the cell that receives the signal? Can all cells react the same way? And the answer is no. So, especially in the case of ligands that are secreted, the cell, in order to be competent, has to express the appropriate receptor, right? So not all cells will react to wingless signaling if they don't have the Wnt receptor there, you know. So you need expression of all components of the signal transduction pathway. So even if the whole embryo at an early stage experiences a signal, perhaps only certain cells there have managed to already transcribe and translate into protein the components of the signaling pathway, right? And then you will need, similarly, the expression of the particular transcription factors downstream of the signal transduction machinery to change gene expression. Or in the case of these nuclear receptors, they also they have to be present, right? So, and then cells can be said to be competent only during a restricted time window. So it can be that this is the same thing, that first you need to have these things present, and maybe they are present in cells at a certain time, but they're not maybe maintained as the cell changes fate in response to other signals, it may lose the competency to respond to the next signal. Okay, so when it comes to these inductive signalings, they don't have to operate over large ranges because they're happening during development when the embryo is small, right? So typically, patterns established by inductive signals are established in small primordia of organs or limbs, or before it gets long and distant. The patterning happens at a very early stage, and then growth of that basic pattern happens afterwards. Okay, and this is just to expand on the previous slides, that the responses to inductive signals depends on the state of the cell. But I want to add on to that the inductive signal, we can have combinations of inductive signals, and that the cell will remember these things. It has a cell memory, and that can expand the repertoire of responses to the same signal. So let's have a look at that. So we can have a case here where two different cells, let's say, signaling to the same cell. In the first case, the cell releases signal A and signal B and gives rise to a cell type that is green, just to show that the inductive cue will lead to a green cell. Signal A or signal B alone might lead to another color, right? And then we have another cell that can signal to this same cell, but it signals with signal A or signal C, and that combination gives rise to this purple looking cell. Okay, so it's not just that the cells receive cues from one inductive signal, they can receive several, and that can be translated into different outputs depending on what the inductive signals are. Then there's also cell memory, where we have one cell signaling to another cell, triggering its nucleus, let's say, to differentiate a little bit so that it has a black nucleus. And then another signal comes in, signal C, and this will trigger changes in the cell that in this schematic cause the cytoplasm to turn green. But if this same cell experienced first signal Y that caused its nucleus, instead of turning black, to turn white as a differentiation marker, and then received the same signal C, it can respond differently because it has memory of the first signal that it met, and these cause transcriptional changes in the cell that change the way it interprets signal C. Even though the signal is the same, the cell type is now slightly different, and it can respond differently and give rise to a cell that differentiates differently. Okay, and this in a way feeds back to our responses to inductive signaling. They depend on the state of the cell. So an inductive signal can select from a response from a small number of alternatives, if it's instructive, the instructive signal, right? And cells in different states. So for example, they might be ectoderm or mesoderm. They have distinct sets of possible responses because they have a memory of what they have met before. They are determined in some way, and they can respond in different ways to the same signal. So in the example given here is sonic hedgehog. When secreted to structures that are determined to become limb buds, this will give rise to, this is actually an inductive signal to trigger limb structure development. Whereas if sonic hedgehog is secreted to the neural tube, which is already, and it's a ectodermal tissue that is developed in a neural manner, the sonic hedgehog signal will trigger it to pattern in a dorsal ventral way its neurons, right? So the response in the different tissues is different, even though it's to a response to the same signal. This is another conceptual model to illustrate that we can have patterning being developed by sequential induction. So in this case, we have a region B that is secreting a signal onto A and induces the formation of region C. And then C, which is different, as a different cell fate, let's say, from A and B, can produce its own signal. And the same signal produced from C will produce different results when it's secreted and induces tissue from A. In this case, it gives rise to D, and the signal from C, when it's secreted to B, will give rise to a different type of tissue or cell type. We're making a different pattern here. So this just illustrates the last point again, that it's the context in which the signal is received that's important. How competent are the cells to respond to that signal? This is perhaps a good time to introduce the idea of morphogens, and they are secreted signals that act over a long range and have a graded response. So what do we mean by this? We have a field of cells and we get the secretion of a morphogen from one side of the cell, and this will give a concentration gradient with the highest concentration of the morphogen close to the source and the weakest away from that. Then the cells can respond differently to the concentration of morphogen received. And this is one way to establish pattern. And we will see this in the next lecture in more detail. It's good to just introduce the idea of a morphogen and that it forms a concentration gradient and that cells can react differently to different concentrations of that gradient. This is explained in a different way here. I'd just like you to read through this yourself. It's something that we will go through in more detail in subsequent lectures. So while we've discussed that this sort of positional information can be a general mode of pattern formation, so morphogens, there is an alternate strategy that's worth bearing in mind, and that's sort of based on differences in cell adhesion, so differential adhesion. And while we've discussed pattern being generated by these kind of models here, where one set of cells starts secreting a factor that affects the neighbors, in differential adhesion, we have mix of cells that start expressing different adhesion proteins on the surface, and these have a tendency to sort themselves out into a pattern. This is used in some instances in development, and it's been observed in transplant experiments, for example. It's just bear it in mind, even though primarily we will be discussing patterns being generated by morphogens and positional information. So many animals develop structures that are regularly spaced. For example, feathers on birds, ommatidia in the compound eye of insects. Even if you look at your arm, the hairs on your arm are typically regularly spaced. And this is achieved primarily by a process called lateral inhibition, which is really worth knowing about, right? So it generates these spacing patterns in fields of cells. So if we just go through it quickly, all the cells within the developmental field are initially thought to be kind of equivalent. They have the same developmental potential, but in general they secrete a low amount of inhibitor, whatever that inhibitor may be. The cells that begin to differentiate early, they secrete more inhibitor and prevent the differentiation of adjacent cells. And then over time, we will get the emergence of a pattern. And this will give rise to evenly spaced structures. And there's some examples given here, which I don't think you need to know, but the delta notch signaling is very important, and I will go into that in a little bit more detail here. And what's important to think about is that, yes, all these cells are sort of equivalent, but in fact, if we turn this previous slide on its angle here, we see that already they're emerging in lines, and there is something known as a pre-pattern that is laid down before, which is these stripes of cells, so that are more equivalent than others. And delta notch signaling between cells is then important to space out the cells more. So the way that delta notch signaling works, I've told you, one cell expresses delta and the other expresses the receptor notch. But in fact, in these cases, both cells express delta and both cells express notch. And they and their surrounding cells try to argue and fight to see who has the highest level of delta ligand expression. And delta expression will try and shut down via notch the production of delta in the neighbor cell, right? So they all express delta and notch, but in the beginning it's thought that within this field, one of them randomly expresses a little bit more delta that causes notch signaling to be shut down in the neighbor cell, and this reduces the production of delta signaling to this cell. And this small difference is amplified over time to give rise to sort of a winner cell and a loser cell. And typically it's thought that the cell that had slightly higher of delta in the beginning wins, and it will signal to its neighbors to shut down. And you need to be a certain distance away to get another delta positive cell winning. And this can be because of the secretion of this of an inhibitor in a certain radius, or the existence of a pre-pattern laid down in stripes. This is complicated. We're getting complicated now, but it can't be avoided. I'm aware of time issues here. So I just put up these slides, but I will skip through them. But just so you know that they're there, they are things that we have covered already. I feel asymmetric cell division, for example, here, and I put them up. The PowerPoint is uploaded as well. You can read through these. I think it's important to mention that the development of embryos should be consistent and reliable. It's a very important process, and the organism invests a lot of energy into making eggs. They should be successful. And of course, reliability is necessary for making proper functioning adults and then passing on the genes to the next generation. So internal and external fluctuations should not perturb development. So, and what do we mean by, well, external? We could take temperature, for example, or humidity. These should not interfere with development. And internal, we mean like slight differences in signaling levels. You should allow for that. You should allow for differences there, but the system needs to be robust enough to survive these small differences that can occur during development. And this reliability and fidelity in creating an embryo is achieved by a couple of ways: redundancy, two or more pathways that can carry out a particular process, and there's a backup then if one pathway fails. Also, though, you have this negative feedback where the end product of a pathway can inhibit an earlier step in the pathway, so that you maintain a certain level of end product. And then are several networks of pathways, and they are intertwined, relatively insensitive to small changes in individual steps in those pathways. Okay, so I will try and quickly go through model systems, and we wanted to start with the relationship between animal models, so that the phylogeny between them, the degree of relatedness. And this figure here is a phylogenetic tree, which is based on morphological and molecular criteria, and it gives us some definitions by which we can cluster animals we consider more closely related to each other, not just on the level of DNA sequence, but on other factors that they have in common. And one of the important ones was radial symmetry, and I described this in the first lecture, where you have basically an organism which is kind of round and has no clearly visible left and right side, let's say, and typically these animals can be clustered by the number of germ layers, and these early radial symmetry animals had two germ layers, so they were diploblastic. These are shown here, and we can see that there are older animals more distantly related to us, for example sponges, while the radial symmetry animals here that we have heard a little bit about before, Cindaria and Centophoria, and some of the guys from the SAR Center were working with some of these, so it's worth just mentioning that these are distantly related. Closer related to us was the event of the bilateral symmetry, so then we can talk about sort of a left-right handedness to that symmetry. It was easier to define up, down, dorsal, ventral, anterior, posterior, right? They have these symmetries, and three germ layers then that we have talked about before. And from here, we can split the animals into large groups on the basis of where the mouth forms in the early embryo. And here we have echinoderm, this a starfish, or is it a sea urchin? I think it's a sea urchin actually. This is from the book, you can check it out. And here we see that the blastopore forms is forming down here, and we get an invagination here that will be the gut, and so the gut or the anus forms first, and then the mouth develops and joins up with the gut. So the mouth develops second, and these are called deuterostomes. Okay, this is a classification on the basis of what forms first, whereas uh, protostomes, they develop with the mouth first. So the blastopore, when it forms and we get the invagination into the embryo, it will be the mouth that is left around the blastopore. So this is a distinction made, and most of the model systems that I have mentioned, for example, flies and worms would be in this protostomes. So they are a little bit different. Here's another, don't worry about that. Different types of invertebrates, these, for example, insects, they can be both protostomes and deuterostomes, but they have a very diverse development. But useful to us is that they have features that are common with vertebrates, for example, the cleavages, the blastula stage, gastrulation, these movements, and they have 3 germ layers. Um, Many of the invertebrates have more mosaic embryos. If you remember, that's these determinants that are distributed early on between cells as they divide, as opposed to regulative development. Generally, they have a much lower number of cells at the gastrula stage, and because they are sort of less numbers of cells, cell fates are in many cases specified on a cell by cell basis rather than in groups. In many invertebrates, development does not involve gradients of positional information. For example, C. elegans, but in Drosophila it does. But we can talk quickly through these model systems. We've mentioned them before, quickly through it. So the nematode model, Cenorhabditis elegans, is living in the soil. The egg size is about 50 micrometers. So that's quite small. They're easy to culture just on agar plates, and they're highly suited for genetic analysis with a short life cycle. We've covered a lot of this before. Special features though, they are transparent, which is very good for imaging. They have few cells and they have an invariant division pattern. So this is something that's very particular to this animal, that they have a cell lineage that is well described. And on the next slide here, I just wanted to show that basically if we take the zygote starting here, I know this is difficult to read and you don't have to read it, but each branch point here is a cell division. And down here we have the terminal cells. And the point I want to show you here, or the point I want to make, is that every division in this animal is known, and the cell fate at every stage of development is known and given a name. And here we have the terminal differentiated cells, and whether the cells live or die, everything is mapped out. So we have a cell lineage for this animal that is very useful to study developmental events. They have a small genome, more genes than in Drosophila. The rest you can flick through and have a look at yourself. But here's some pictures of the embryo, and we'll meet this later on as well. You can visualize it with a light microscope. It's very convenient to work with. And the insect model Drosophila, I've talked a lot about this. It's probably the best understood developmental system. It does rely on positional information specified by gradients. So we get this specification of groups of cells, just like in vertebrates. So this is very useful to study that. Special features, an oocyte with yolk in the center that has a pre-established anterior, posterior, and dorsal ventral polarity. So these are already set up in the egg before it's fertilized. One of the things I would say is pretty cool is that it's a large egg considering the size of the insect. The egg is about 0.5 millimeter and can be seen with the naked eye actually, even though we use a microscope to manipulate it. It has a syncytial blastoderm, and if you remember, a syncytia is a large cell that has many nuclei in it. And I mentioned this when it came to muscle fibers, where the cell walls had broken down between the cells, and we have a syncytia. And the same is true for the Drosophila egg. When it is born, we have this very large egg with the single nucleus, and this nucleus begins to divide without having cellularization in the embryo. So we get many, many nuclei in the cell, in the large cell, and then it's only later the nuclei move to the periphery of the embryo, and then we get cellularization. Okay, this is sort of indicated here. In the syncytial blastoderm, actually, we can have gradients of transcription factors, and we'll see how this works later on. The embryo becomes segmented, so we can study how segmentation works in this animal. And then it has imaginal discs, which have true metamorphosis, like insects have, where we get new adult structures from tissues that are developing during the larval stage that then, through metamorphosis, become the adult. Just going quickly through this. Amphibians, the frog, Xenopus laevis, the eggs are large, 1 millimeter, so double the size of the fly egg, but the animal itself is obviously much bigger. So, but they can give rise to large numbers of fertilized eggs. They're easy to obtain. The embryos are resistant to infection after microsurgery. That's why they can do these things where they remove one cell after the first cleavage. It's also easy to visualize, so that's quite nice as a model system. You can also culture fragments of embryonic tissue in cell media. But there's a huge advantage. I mentioned that already. They're tetraploid, so they don't have a diploid cells. They're tetraploid and they have a long generation time. And these two things argue against using it as a genetic model. So birds, the chicken has been used. The eggs are easy to obtain. Mammals and birds have very similar morphological features, and the gastrulation process is similar, so we can use them to study that. And they're a valuable supplement to mouse embryology. They have the same features as human and mice, like the development of somites, the brain and limb development can be studied in chicken. They have a small patch of cytoplasm with the nucleus on the yolk surface. And then they have this primitive streak that forms, shown here, which is very similar to mammals, so we can use the chick to study these things. In mammals, we have the mouse model, and they have a sort of short-ish life cycle. I wouldn't argue that this was too short. It's 2 to 3 months, and they're relatively easy to keep in the laboratory. They're a good genetic model with mutant screens and targeted knockouts being possible. But I mentioned this before, development is the first stages of development are inside the mother, so you can't manipulate and observe as it's developing. The eggs are small, so 0.1 of a millimeter, so 5 times smaller than a fly egg, and that is not a good thing. And they're protected by an external coat, so they're not that easy maybe to manipulate. Yeah. And I just put up this picture of, so this embryo is seen in 2 directions, and this is just in one, but you can see how similar these pictures are. And one of these is mouse and one of them is human, but I don't remember which one is which. And that was the kind of the point, that they look so similar. They're really a good model for studying human development. Fish, we can't go anywhere without mentioning fish here. So the zebrafish model is easiest to keep in the laboratory. It does take a lot of space with the tanks, with the water, but relatively easy. They have a relatively short life cycle, about 3 months, so about the same as mouse. And they give a large number of transparent embryos, and that's really a plus, that you can get so many eggs, and you can control the fertilization of them. And these can be used for efficient mutant screening. Okay, sorry. So I had to rush there. I realized that the whole lecture is getting too long. We're approaching the 2-hour mark in total, which I think is just too much. So I went quickly through it. Most of this introduction to the development of model organisms you've heard before, and you will hear when we start now next week. It'll be with Drosophila development for a couple of lectures, and you will hear a lot more about it. So I just give you a quick primer for those oncoming lectures. But other than that, we have discussed pattern formation. I hope that you are starting to get a grasp on that, and we will go into more detail on that. You can use the Zoom meeting to ask me more questions there if you want. These sort of terminologies and stuff is just to try and understand and learn definitions of them. And again, we can discuss it if you want. And then it's good to know the techniques that are used to monitor developmental genes. Okay, sorry for going so long. Thank you for your attention. See you at the Zoom meeting. Bye.